Okay, can you hear me? Okay. Thank you very much, Erica. So as a biologist, I've always been particularly interested in understanding how cells move. And this is a very critical part of disease because, as many of you may know, most cancer deaths actually happen when the cancer cells leave the original tumor and travel to other organs in the body. And what I'll show you here in this movie on the right is a tumor with all the cancer cells shown in green. And what you might notice about this image is that most of the cancer cells are actually going to remain stationary in the tumor. And that's where they'll stay. Oops. But sometimes, some of the cancer cells can undergo a transition from a stationary to a mobile cell type. And this is a process we call EMT. And you can see that here with these two cells that have become mobile and are able to escape the tumor. And once they start moving, they can find and enter a blood vessel, shown here in red, and then travel through circulation to other parts of the body. So cell migration is really an important and deadly aspect of cancer, but it's also a really difficult thing to study because normally we can't see through humans or mice to be able to watch these cells moving. Our microscopy techniques are getting better, and we're able to take these short little movies by making a little window in the skin of an animal. But there's a lot of technical challenges with doing these type of experiments, a lot of limitations to this method. Another way would be to take these cancer cells out of an animal and put them in a petri dish and watch them move. But we know now that cells migrating on a flat surface just does not well represent how they migrate in a complex three-dimensional environment of the body. So an alternative approach to study cell migration is to turn to a model organism. And it turns out that sea urchins, that you can see here, actually provide a really fantastic model system to study cell migration. They make these beautiful embryos that you see here in the top images. And they develop outside of the adult animal, so we can watch them develop. And they're completely transparent, so we can just watch them under the microscope developing over time, just like you see in these images. And what's also interesting about the sea urchin embryos that you can see in this bottom images is that they have a group of cells here that are shown in green that are very similar to cancer cells in that they undergo the same process of EMT that makes them transition from a stationary to a more mobile cell type. And so they start out as a pool of cells at the bottom of the embryo, and as the embryo is developing, they migrate up the inner wall of the embryo to form this ring structure. And once they're in the correct location, they start to synthesize components of the skeleton. And you can see that here in this older embryo where the skeleton is shown in white. And if these cells don't get to the right spot, then the skeleton won't form properly. Now, we know that these cells do not inherently know where to go on their own. And they're actually receiving some type of directional cues from the rest of the embryo that help them navigate and figure out where they're supposed to go. But what these cues are that help them navigate is unknown, and that's what we're trying to figure out. So something else that we know is it's not just cells that are migrating on the inside of these embryos. If we use a special type of microscopy, you can see the inside of the embryo and find that just like tumors, these embryos are actually filled with a network of long and thick protein fibers. And in other instances, these types of protein fibers are known to act as tracks for cells to migrate along. But our question here is, what proteins are these fibers actually made of? And are any of these proteins in this fibrous network providing the directional cues that help these cells figure out where they're supposed to go? So to answer these questions, we treat normal embryos that you can see here up in the right hand corner. We treat them with a special detergent that can remove all of the cells from the embryo. And after we treat with the detergent, what we're left with is what you see here in my image that's in the gallery. They appear as these glassy membranes that are surrounding the inner skeletons. And these glassy membranes are made up of this whole fibrous network. So once we've isolated these fibrous membranes, we can study them and figure out what the proteins actually are. And once we have that information, we can go back to the embryo and do some cool experiments where we genetically remove some of the proteins from this fibrous network and then ask, can the cells still move without that protein there? And if so, have they lost the ability to navigate to the correct locations? So what I'd like you to take away from this talk tonight is that even though sea urchins are evolutionarily very distant from humans, they're still a really powerful model organism that we can use to study the biology of cell movement. And this can give us really important information about how cells move in order to form an embryo, and also how cells might be moving during cancer metastasis. And so with that, I'll thank you for listening. Thank my advisor, Richard Hines, and our collaborators, both at MIT and the Broad and BU, in our funding sources. Thank you.